Hello, everybody. My name is Louise Venny, and I'm the chair of the Wildlife Toxicology Work Group. We are an organization that's 60 to 70 members, and we're a wide variety of practitioners and just interested folks. Today, we're excited to share with you the webinar about PFAS. These are perfluorinated compounds, and we'll learn a lot more. Today's purpose is to reduce how much chemistry we actually have to think about, but actually look at how things affect wildlife. And so we have four great speakers for you today, where we'll start out with Amy Quinton, and then we'll move on to Chris Custer, Mark Johnson, and Sean Strom. So with that said, Amy, if you want to get your slides up. Amy is a human health risk assessor at Wood, and her role is to lead and review multimedia human health risk assessments for common and emerging contaminants under state and federal programs, as well as internationally. She's been leading human health risk assessment work for PFAS at Department of Defense sites for several years. Amy.
Thanks, Louise. I'm going to go through some very highlights, let's say, of PFAS. We'll talk about where they come from, how they might enter the environment, and some very brief highlights about the chemistry. And then I will turn it over to my other colleagues to talk a little bit more about the wildlife elements. So without further ado, our introduction to PFAS. Next slide, please. So we will talk, as I said, a little bit about the chemistry, fate and transport, and then we will touch on some complexities about sampling PFAS, and then we'll start to get into a little bit about the toxicology. As Louise mentioned, I am a human health risk assessor, so I will have a slide or two about how this affects humans as well. Next slide. And next slide, please. So we'll just start with a brief history of PFAS. So you will have heard PFAS in the news recently, but it's actually been around for quite a while. So these are synthetic chemicals that were first synthesized after World War II. So we see, they first start to enter usage around the 1960s, but they get much more widespread in the very late 1960s, early 1970s. So this is when the FDA approved their use in food packaging, and DOD issued what they call a mil-spec for AFFF. So that's aqueous film-forming foam, also known as firefighting foam. PFAS, because they are they're synthetic chemicals that have very unique chemical structures. So they turn out to be extremely useful for certain things, like fighting petroleum-based fires. So this AFFF, firefighting foam, became very commonly used in airports, petroleum bulk storage, those applications. And because PFAS has amazing stain resistance properties and other properties that make it very useful to impregnate fabrics with. We see use of it in carpeting, in clothing, in anything essentially that could be stain resistant or water resistant. So by the late 1970s, PFOA was detected in the blood of workers, and by the 1980s and 90s, it was detected in drinking water. And it started to be recognized that these were persistent organic pollutants. And by the early 2000s, certain PFAS chemicals started to be phased out. So PFOS is a specific chemical that we're going to talk about more as we move along, as is PFOA. And so basically by the late 2000s, we see that the PFOS and PFOA production has essentially ceased in the United States, not necessarily everywhere, and levels across the population have started to drop as far as what's detected in the blood of humans. Next slide, please. So some common products that you might see PFAS could be in: food packaging, as we said, any stain resistant fabrics. So this would also be water repellent clothing, as well as fabrics that could be in carpeting or other upholstery. And then this aqueous film forming foam. Next slide, please. So one of the main facilities that we look at this point is like fire training areas. We tend to see them on military bases or around airports. where you might have had this aqueous film forming foam repeatedly used in the same places. Anything around an airport where you might see a crash site where there might have been a large release of AFFF, that could also, be a release that then ends up moving into groundwater or surface water. There are other facilities where AFFF can be an issue, or other types of PFAS. So for example, metal plating, finishing facilities. And then there's also landfills. So PFAS can be in wastewater treatment plants, ends up in biosolids, and those of course can be applied to agricultural lands. So there's a lot of different ways that PFAS can enter the ecosystem and the environment. Next slide, please. So were you to come across some bulk storage of AFFF, this is essentially what these canisters would look like. Sometimes when they're stored like this, they can be corrosive, they can leak, and they can be released right to the groundwater, right from their storage containers without ever being applied. So that is another possible issue where these storehouses still exist. Next slide, please. So we're going to talk very briefly about the chemistry of PFAS. Next slide. So first of all, when we say PFAS with an AS, this is talking about a very big class of chemicals. So there's over 3,000 different PFAS chemicals that have been identified at this point, of varying lengths, varying amounts of fluorination. But when we talk about looking at PFAS on a specific site, we're usually talking about a much smaller group within this 3,000. So most of the analytical methods right now are looking at around 20 PFAS chemicals. They can look at more, they can look at 70, 100 potentially, but typically standard investigations are looking at about 20. And within that, there's not a lot of toxicity information for most of those chemicals. So for human health, we're really looking at about 3 chemicals that we have toxicity information for. So although there's a very broad class of chemicals when we talk about PFAS, there's really only a few that we're able to evaluate and look at how A, how prevalent they are, but then B, how they affect people and the environment. Next slide, please. So what does PFAS mean? So it's poly and perfluoroalkyl substances. So they're alkanes, which means they're carbon chains, and they're fluoro, so they're fluorinated. So we're looking at fluorinated carbon chains. And this fluorine-carbon bond, this is one of the most, the most persistent and strongest chemical bonds that there is. So these are very hard to break down chemicals. And they can be varying lengths. So we see here it says C3 to C20. So these are different chain lengths of these alkanes. And they tend to have very specific structures. So their head and their tail, as we call them, have different properties, which is why these chemicals were so interesting to manufacturers and so useful, because they were able to, this tail has this hydrophobic and lipophobic qualities, which means it resists stains and it resists water, essentially. So they were able to use these chemicals so broadly because of those properties. And so we're going to talk a little bit more on the next slide about what poly and perfluorinated actually means. Next slide, please. So the polyfluoroalkyl substances, these are what we typically would call a precursor. So it basically means they're not fully fluorinated, but they could become fully fluorinated. So when they're fully fluorinated, that's when they reach their final state. That is their final, most resistant to breaking down chemical structure. So on this first column, beneath the 2 bullets, there is a big chemical structure. You don't have to pay too much attention to it, but it is just an example of a precursor, and that's a precursor for PFOS. So on the right side, the right column, that first colorful one with the tail and the head is the chemical structure for PFOS. And if you look at the chemical name, it says perfluorooctane. So we know it's a chain of 8 carbons long. And it says it's a sulfonate, so we know what the head type of that chemical is. The perfluorals, they tend to be sulfonates or carboxylates. And as you can see, there's an example of both. And for those that have heard of the health advisory for drinking water, it's based on PFOS and PFOA, which is these two chemicals here shown. And you can also see that the head of these has a negative charge. So these are also anionic chemicals, which is another reason, they have the quite interesting and different kind of chemical, qualities. So anyways, this is why, we call these forever chemicals, because they're very persistent. And once they reach this final form, they're very, very hard to break down. Next slide, please. So we'll talk briefly about the fate and transport. Next slide. And so we can just click through. There's a couple moving parts to this slide. So this is a conceptual site model for a fire training area. And so in the first couple of circles that have just shown up, we can see that it is surface dispersal typically. So it's a foam. So it gets, dispersed either on in a crash or in a fire training area. And from there, it can either move across the surface into surface water bodies, or it can move downwards into the groundwater. And PFAS can be very mobile. So, it will move readily into these water environments. And it's worth mentioning on this one that the surface water, this is really where we see PFAS entering the food chain and interacting with wildlife. So we either have aquatic animals in the surface water bioaccumulating these substances, or we have terrestrial animals drinking the water and just being repetitively exposed where there's a source area nearby. PFAS can travel pretty efficiently in groundwater and in surface water. And so we can see impacts to both surface water and also residential drinking water wells at quite some distance from the site. And I also want to mention that we see this a lot in surface water, in freshwater. But once we reach a marine environment, what we've seen is that at least PFOS and PFOA tend to drop out a little bit of the solution, possibly because of that anionic charge. When they reach the seawater, we have seen less bioaccumulation in those areas. So even though PFAS can be found across the globe and in very, far distant from particular source areas, the bioaccumulation in those marine areas doesn't seem to be as prevalent as in the surface water environments. And I also want to mention that there's a lot more information. This is just very much touching on, what's out there. And if you're interested more in the chemistry and the fate and transport, there's a link at the bottom of this slide for ITRC, which has a lot of great information as well that's available. Next slide, please. So sampling for PFAS can be a little bit challenging. One of the reasons it's challenging is that we're looking for very tiny amounts of PFAS. So the health advisories are around a drop of PFAS in an Olympic-sized swimming pool. So it's a very tiny amount of PFAS that we're looking to find. And you add that to the fact that PFAS in general are still quite ubiquitous. So even though certain chemicals have been phased out, PFAS as a class is still being, produced and used, and it can be found in a lot of consumer products to this day. So even small amounts could potentially cause cross-contamination in sampling when you're looking for very, very low amounts. So specific sampling protocols are used to try to manage this. Next slide, please. So just a couple of examples. When we're sampling or when lab equipment is being used, we try to avoid products such as Teflon, which may potentially have PFAS in it. And it's not alone. There are definitely others. And then, looking at the consumer goods, potential equipment that you could be wearing, for example, a water-resistant jacket or fast food wrappers. And there's a lot of discussion going on and some research going on to find out, what's really likely to drive cross-contamination and what's not. If someone uses the wrong shampoo before they go out and sample, is that really going to have enough potential PFAS in it to change the results? Probably not. But it needs to be kind of documented. And so until that's fully done, we do follow very strict laboratory and sampling hygiene protocols. whenever PFAS is being sampled. It's just something to be aware of as we move on. Next slide, please. So I'm going to touch very briefly on the toxicology and risk assessment concerns. Next slide. So the real issue, at this point, and what's been driving, a lot of the research and why PFAS and PFOA have been in the news recently is because there is a dose-response relationship with health effects and taking in PFAS and PFOA. So essentially, you're exposed to it, typically through ingestion, and if you're exposed over certain levels, then there are potential health effects that could be related to that exposure. And some different health effects that have been associated with PFOS and PFOA. And again, we're talking specifically about PFOS and PFOA now. We're not talking about the entire class of PFAS, as there's ongoing research, but it's still very much emerging for the others. But for PFOS and PFOA, particularly, there's effects related to pregnancy. So these skeletal variations, delayed ossification of the phalanges, I believe, is one of them, which is, it's questionable that there's a long-term effect there, but it is an effect. And there are other effects which, have the potential to be serious, such as cancer, effects to the immune system, thyroid effects, and these potential cholesterol changes. And these, they've been noted in the laboratory, and a lot of them were also noted in this C8 project, which is an epidemiological study that was done, which also indicated a few additional, potential effects that were correlated with exposure to PFOS and PFOA. So there's very limited information still from other pathways. So most of the work that's been done so far is focused on the ingestion route of exposure. Much less research has been done on inhalation and dermal exposures. Next slide, please. So when we do look at human health exposure, drinking water is generally the first initial and main exposure pathway. We all drink a lot of water every day, and PFAS is very mobile and ends up in the water. So if it ends up in the water and you're drinking that on a daily basis, there's a pretty long half-life with PFOS staying in the human body, and so it has the potential to bioaccumulate. But running a second place in the, what we evaluate for human health is then, can PFAS enter through the diet? And that's where you're potentially going to run across, how you might find, if you're, say, near a military base, that, suddenly the Army wants to sample fish or game in the area, or is looking into these other pathways of how PFAS can get into wildlife, and then from there get into the human food chain, because that is definitely a possible pathway. And then we see these lower exposure pathways. It's possible to be exposed to PFAS if it's still in the dust. As we said, it's a surface application. So potentially, PFAS could still remain in surface soils. But it tends to be in areas that are either still industrial in use, or otherwise it is a lesser exposure pathway. And then we get down to dermal exposure and inhalation. These C8 chains are pretty heavy chemicals, so they're not volatile. So typically, if there was inhalation exposure, it would be through dust, which tends to be a pretty low exposure pathway when you compare it to the amount of drinking water that someone ingests in a day. Next slide, please. So PFAS is in the news, and you're always seeing these, alarmist articles about forever chemicals, and it's obviously a concern because they're emerging contaminants. There's a lot of uncertainty. Where is that dose-response relationship? How well do we understand it? Are the levels that are currently being regulated to protective enough of human health? There's a lot of open questions, and people are, quite rightly concerned about it. So what we're seeing is that in many cases, these community concerns are driving a lot of the actions. And certain, let's say, the traditional way of looking at a site is to understand the source area and then move outward. But with PFAS, because this is so you know, within the community already, and because it's in the news already, you're seeing there's might be earlier sampling of sort of those final exposure pathways. So looking at, you know, fish really early in the process instead of later in the process. So it's kind of pushing forward some of the animal and tissue sampling earlier than we would maybe have seen it on a different type of chemical site. Next slide, please. And so this brings us to risk to the environment. So ecological risk assessment, it's a little bit behind where the human health risk assessment is, but you know, the research is ongoing and continuing. And in the same as human health, PFOS and PFOA are typically the chemicals where there's more information. Bioaccumulation and biomagnification are certainly relevant issues. But we've also seen cases where we have found PFAS in fish tissue, but not found it detected in surface water, which is quite an interesting issue, whether it's lower detection limits needed for surface water or half-lives in fish, not sure, but there's definitely ongoing research in that area. But typically the approach at this point is to try to collect site-specific tissue data, as opposed to trying to do exposure modeling, which is exactly the same as for human health. Our typical exposure models weren't created for PFAS, and they don't tend to work very well with PFAS. So the predictions can be very off, and they can be off in various directions, whereas when you actually collect tissue data, it gives you a very accurate sense of what's happening. There's no US EPA values at this point for the aquatic life and terrestrial criteria, but the state of science is changing very quickly. So, you know, watch this space. Next slide, please. Okay, that's my highlights of PFAS. I hope you found it helpful, and I will hand it back over to Louise.
Mark Johnson is currently serves as Director of Toxicology for the U.S. Army Public Health Center at Aberdeen Proving Ground in Maryland. And it's at that location that he's responsible for the operational and technical arm of the Army Surgeon General and the Assistant Secretary of the Army for Toxicological Matters. He's worked extensively in the evaluation of toxicity of military unique compounds and development and evaluation of a phased approach to gathering toxicity data for new compounds under development. So you can see why we ask him to speak. Mark, all yours.
Hi, thanks, Louise. Get right to it. First, common disclaimer, what you'll hear from me today are my ideas and not necessarily those of the Army or Department of Defense or the federal government. And I want to thank, right off the bat, Ali Narazano, Mike Quinn, Mark Williams, and all the folks back at the Tox Directorate, because a lot of the stuff that I'll show you today is a result of their work. And so I'm going to repeat a little bit, not too much, of what some others have said before. PFAS really is a universe of different molecules. Typically, the longer chain molecules are going to be a little bit more resistant, a little more recalcitrant than the shorter chain molecules. You'll hear a lot of PFOS. You just heard of PFDS, but there's some other ones as well. In fact, as was heard before, there's over 5,000 different forms of per and polyfluorinated alkyl substances. And in fact, a lot of ways these things can branch off. They can actually resemble fatty acids in a lot of ways. And they tend to act like that. They seem to act like that in the body, in the liver. They may interculate basically into cell membranes. And actually the role of fatty acids in a lot of functions in humans as well as wildlife are relatively unknown. They tend to play a role in immune function, which kind of makes sense because some of the effects we're seeing are a result of immunosuppression. And I'll talk a little bit about that in a few minutes. Some of these, because they're halogenated organics, are incredibly stable. I mean, we learned this from PCBs, right, and dioxins and other molecules. But the difference here is those molecules tend to partition to fat where these really don't. They don't seem to. They tend to have this hydrophobic side and this hydrophilic side, right? Almost like, well, they're emulsifiers. They're designed to be that and to actually squash oxygen consumption and to squash fires in a lot of ways. But they do a lot of things. They're excellent surfactants. They're very difficult to do aquatic toxicity studies with because they're really hard to get into water, homogeneously into water, at least in higher concentrations. They want to float to the top, right? And in vivo, they're associated with albumins and proteins in the blood. So there's a lot of protein partitioning of these compounds. And as I mentioned, a lot of times they're associated with bioaccumulation in the liver. And like Amy mentioned before, they come from a variety of sources. And I really like what Chris did, because what she showed is that in a lot of ways, we're closing this barn door after the cows are out. This thing happened a long time ago. They stopped making some of the more recalcitrant ones, and that's good. But we're still seeing things, right? We're still seeing effects and exposures almost circumpolar. There's also, as I mentioned, there's the fluorotelomere alcohols. I mean, we're talking 5,000 different types of molecules. There's the polymers that really, some of those aren't very recalcitrant at all. They're very much in flux. They don't last very long. They tend to form and reform. And so it's really difficult to talk about these molecules as a class. It really is. I think the only thing they really have in common is their organic molecules, alphatics with fluorine. And we're finding these things all over the place, as I mentioned. We're finding them in tissues of aquatic organisms from the Arctic. As was mentioned before, the NHANES study, which is from the CDC, the National Health Nutrition Examination Survey, found that 98% of us have some of these molecules in us. Is it from Gore-Tex, from Teflon use in pans, Stainmaster carpet? Who knows? But this stuff is really everywhere at this point. And also, we've also found that it is associated with drinking water, at least some of the potential effects, more in this country than other places in the world. But there are some other places in the world that are thinking really heavily about biosolids collected from wastewater treatment plants, and how those solids are being used to reconstitute soil in some of our grazing fields. Particularly Australians are very interested in that. And so there's lots of sources that we're looking at in addition to that from drinking water. And so species with detected tissue concentrations of PFOS, perfluorooctanosulfonate, include a lot of gull species. Like there's some listed there for birds at the top, seals, marine mammals, including polar bear, mink, brown trout. I'm not going to read them all, but you get an idea. As I mentioned, it really is quite circumpolar. And to give you an idea of the concentration that they're seeing out there, this is nanogram per gram wet weight. Gives you an idea of what concentrations are out there. Birds tend to have some fairly high concentrations in plasma, but these are different tissues, right? And so we need to think about that as well. And so another way to look at egg PFAS burdens, these are right here, just like 3 different molecules of different PFAS to include PFOS, PFNA, PFO in some circumstances. Each graph varies a little bit. And you can see how some concentrations for some of them are going down, but some are going up. And just like what Chris was talking about before, PFUDA, for example, is going up in auklets. In petrels, it is as well. But PFOS concentrations seem to be dropping. PFNA seem to be going down a little bit too in some birds. So I'd like to talk a little bit about toxicity. We talked a little bit about the fact that this stuff seems to bioaccumulate in the liver. So a lot of individuals are looking at liver. Certainly histologically, it shows up in the liver as vacuoles. What exactly is in this vacuoles is assumed to be some PFOS, but also could be some lipids. It's difficult to tell exactly. We don't have good immunohistochemistry for what exactly is the compound within the vacuoles. But we have seen pretty good concordance with immune effects. We see this in laboratory animals. We've seen some evidence of this in humans. And so the evidence is pretty consistent that the immune system may be affected. But I want to talk about that a little bit more in a few minutes. There is some evidence in laboratory animals that it may cause tumors. There certainly is reproductive effects for some molecules, certainly not all of them. And there may be some evidence that there's some hormone disruption. We talked about the thyroid earlier. And so work in our lab, we've been working with Peromyscus, which are white-footed mice, and deer mice, actually both species. We've seen, like we've seen in other laboratory rodents, some changes in liver size and total liver size for PFOS at different concentrations. You'll note that the unit here is milligram per kilogram body weight per day. That is pretty much the unit of measurement if you want to do risk assessment. If you want to understand what exposure could be, if it's how much is too much, we've heard others talk about understanding what that threshold is. That's typically the units you see it in, milligram per kilogram per day. And then you can model exposure on one side. And of course, if the concentration is higher than where the level at which you start seeing adverse effects in this diagram, it looks like above 1 milligram per kilogram per day, then you can expect, in PFOS, you can expect to see changes in fertility and pregnancy indices. For PFNA, though, we don't see that. So this diagram basically just shows how these two molecules are a little bit different in terms of seeing changes in reproductive performance in Peromyscus. In terms of litter loss, is another way you can look at these data. Again, we see statistical differences at 5 milligram per kilogram per day for PFOS. Repeated dose studies for PFNA, not so much, at least not in Peromyscus. Lots of variability. But what we do see in PFNA is a nice dose response on plaque-forming cells. And what those are B cells. They're tuned up B cells. And this is an interesting way, a good way to look at changes in both cell-mediated and humoral response to an antigen. And so basically you expose these animals to a foreign antigen, you expose them again a couple weeks later, and you look at their response. And if you see a decreasing response related to exposure to a molecule, then that tells us that these animals are immunosuppressed, right? And so we see evidence of immunosuppression in not only our Peromyscus, we see it in mice, we see it in laboratory rodents, and we have some evidence I mentioned before in humans in terms of correlating concentrations of some PFOS in the serum to that with antibodies from certain vaccines. And so there's some evidence for immunosuppression. But however, there is some also evidence, epidemiological evidence of ulcerative colitis, right? And maybe asthma, which are not immunosuppressive events. They're immunoenhancement events. Just the opposite. You're going in a different direction. And so I think those endpoints need to be further explored. Certainly the immune system is a very complex, very well-tuned system. There could be some immune dysregulation going on, not quite sure yet, or there could be artifacts. We'll see. And so typically what toxicologists do is we try to characterize it in terms of what is the no adverse effect level and what is the lowest observed effect level. And what this diagram here shows you, shows us that the effects we're seeing in Peromyscus are almost in the same ballpark that we see in the typical laboratory mouse and typical laboratory rat for both of these chemicals. So we don't expect Peromyscus to be any more sensitive or at least less sensitive. But one thing we found out is that mice are not humans, not even close, especially when it comes to the kinetics of these molecules, the absorption, distribution, metabolism, and excretion. And in fact, in a mouse, the biological half-life in serum is an order of days to weeks. In humans, for some of these molecules, it's years. And so what's going on? And what we found out so far, it has to do with kidney reuptake. The kidneys are different. And the molecules in the kidneys are different that are responsible for the reuptake of some of these molecules. And so I would expect wildlife species to be the same or be similar. And I guess the point of the slide more than anything is kinetics are really important. It's just not the effects. It's how well these molecules are absorbed, distributed, metabolized, and really essentially excreted. And that's what this slide again shows. In rodents, it's 7 to 20 days. And in non-human primates, monkeys, again, it's pretty quick, but not so much for humans. It's around 2 years for some of these molecules. For ruminants and for hindgut fermenters, it's pretty quick. It's very quick, which is interesting to me. In terms of the dynamics side, we try to develop models to help us better extrapolate some of the data we may see in controlled laboratory animal studies to humans, for example. We can do this for wildlife too if we had the data. We certainly do for humans, and they're called PK models. And if when you do that, you learn that the level where this molecular marker PPAR alpha is induced things start happening, both in rabbits, and I'm sorry, rodents, and also in humans. And so that may be what's going on there. There also has been some studies in bald eagles, which maybe we'll hear about later. And so here's some more data that shows some differences in elimination and half-life of PFOA, for example. Again, it's markedly different. In fact, there's big changes between males and females as well. Females tend to be a little bit better at excretion, but not always. So where do we go from here? As I mentioned, the list keeps growing. Last I heard it was 5,000 plus chemicals. It may be up to 8. I don't know. It's hard to keep track. I think it'll help us a lot if we are able to refine these physiologically-based pharmacokinetic modeling, because kinetics is incredibly important with these molecules if we're going to understand how much is too much. I think PPAR alpha is a good place to start in terms of where adverse effects start happening and why. And once we can do that and look at the differences in PPAR alpha affinity between species, we can better extrapolate between species and be able to do a better job at predicting what's going on. As I mentioned, I'm not quite clear if it's immunosuppression we should be concerned about or dysregulation. I'm more on the immunosuppression side because I'm not very convinced on the dysregulation, but we'll see. Certainly there's developmental reproductive effects in mammals and thyroid liver effects. And I think a top-down approach will help us inform, and certainly risk assessment and understanding of what effects could be between species better. And I'm going to stop right there. Thank you.
All right, thank you, Mark. So Chris, if you've cleared that frog, just to note there's a couple questions in the chat and the Q&A, and I assume we're going to get some others coming up here for Mark's talk.
Okay, our
next, thank you. Our next speaker is Sean Strom. He is with the Wisconsin DNR, and Sean earned a bachelor's in environmental science and biology from UW Green Bay and a master's in environmental toxicology from Colorado State University. He has been with the Wisconsin DNR for over 20 years, serving as a toxicologist in both the wildlife management and the fishery management programs. And Sean has studied both legacy and emerging contaminants in a wide variety of species, including fish, bald eagles, water birds, songbirds, waterfowl, fur bearers, and small mammals. If you have other ideas for him on species, I'm sure he'd entertain that too, right, Sean?
Absolutely. All right, thanks, Louise. So what I'm going to do today is kind of take all the other presentations and relate them to kind of a state natural resource monitoring perspective. My slides won't advance. Oh, there we go. So I'm not going to spend too much time on the toxicology of PFAS because that's been done well by Amy, Chris, and Mark. But the primary endpoints are, endocrine toxicity, reproductive toxicity, immunotoxicity, and then, we're still learning a lot about the impacts of these chemicals on wildlife, and so still evaluating potential impacts. So I'm going to talk about a program that was developed in Wisconsin back in 1990 called the Wisconsin Bald Eagle Biosentinel Program. What this program does is that it uses nestling bald eagles to assess trends in environmental contaminants. So a different population or a different region is sampled on a rotating basis, and we collect and analyze plasma, whole blood, and feathers for both legacy and emerging contaminants. The plasma is for all the organic contaminants, whole blood for metals, and feathers primarily for mercury. And what this project does is it provides a snapshot of contaminant exposure in Wisconsin wildlife, which can be useful for multiple agency programs. So these are the populations that were sampled between 2011 and 2017. So our focus areas were the Fox River in Northeast Wisconsin, the Bay of Green Bay, Lake Michigan. In northern Wisconsin, those green dots represent the Northern Highlands ecological landscape, which is essentially a control area. And then the other colored dots, the orangish color, the turquoise and the purple are all on the Wisconsin River. And you'll kind of notice that there's a big gap kind of in the northwest part of the state. Well, a colleague retired with the National Park Service, Bill Rowdy, actually covered those areas using the same protocols, focusing on the Lake Superior populations as well as some of the major river systems in northwest Wisconsin. So what did we find? Well, expectedly very low levels in the Northern Highlands, which is it's an unindustrialized area in northern Wisconsin. Fairly similar levels in Green Bay and Lake Michigan, but rather high levels from the central part of the Wisconsin River all the way down to where the Wisconsin River flows into the Mississippi River. So much higher levels in those populations. And then kind of taking a page from what Chris talked about, that maybe PFOS or PFOS may not be the driver in terms of impacts. These other three PFAS compounds, PFDA, PFDOA, and PFUNA, they were also detected in 100% of our individuals. And some of the patterns are unique, where the levels of PFDA and PFUNA are somewhat similar in terms of the Great Lakes and Wisconsin River, which are both, quite heavily developed, have relatively similar levels. And another interesting observation is that the levels of PFDOA mirror almost identically the pattern that we saw with PFOS or PFOS. Again, we don't really know what the impact of these are, other than that definitely the Wisconsin River eagle populations are being exposed to fairly high levels of these compounds. So just to take a quick turn away from PFAS, just some other emerging contaminants that we've detected as a part of this program. Bisphenol A, which is in the news a lot. These phthalate compounds, I have monobutyl phthalate highlighted. Monobutyl phthalate is a breakdown product of dibutyl phthalate, which is a plasticizer compound used in numerous consumer products. And we detect that in nearly every individual. And then also some very common antimicrobials also show up in these eagles. So what does this mean in terms of management of natural resources, management implications? Well, the results provide a snapshot for resource managers about the fate of contaminants in an ecosystem. So we have very broad spatial coverage. And so, we want to figure out, what do we know about contaminant XYZ? We can say, well, this area has high levels, this area has low levels. Eagles are representative of other species in the ecosystem. They're apex predators. And so, everything below them is likely being exposed to the same type of compounds. This project is very popular with the public. People love eagles, and so this project has a lot of public support. That picture was taken, the nest was in a public park, and as soon as they saw our crew come up, we instantly drew a very large crowd, and the general public is very interested in what we're doing and the results of this type of monitoring. And finally, the results indicate that eagles are a very valuable and accurate indicator of ecosystem health, which can be useful for multiple agency programs. And I'll give you a specific example of that. As I mentioned, the middle Wisconsin River population, which showed very high level or relatively high levels of PFOS compared to other areas of the state. When the fisheries management program with the Wisconsin DNR found out about this, they thought maybe we should start looking at levels in fish in this area. And lo and behold, the fish in some of these impoundments also have rather high levels of PFOS, which resulted in fish consumption advisories. And so this wildlife monitoring actually influenced the fisheries program. And the fisheries program will be looking at fish levels of PFOS in these impoundments for several years into the future. So just to summarize the bald eagles, detected in every bald eagle population that we sampled. From what we know right now in terms of thresholds, the populations are below proposed thresholds, but individuals may be above in certain locations. We've shown that wildlife are excellent indicators of ecosystem health and ecosystem change. And finally, something I usually like to stress is that we are exposed to all the same chemicals that these eagles are. So I'm going to switch gears a little bit and talk about PFAS and white-tailed deer. We have a site in Northeast Wisconsin that produced AFFF foam and is heavily contaminated. There was public concern regarding possible contamination of wildlife, specifically white-tailed deer. Much of this is in relation to some of the monitoring work that the state of Michigan has done, and where they actually have a do not eat deer advisory at a specific location in northeast part of lower Michigan. So in February of 2020, we collaborated with USDA Wildlife Services, collected 20 deer, and from these deer, we collected muscle, heart, and liver samples. And what we found was that we found very low levels or non-detects in the muscle and heart tissue. However, the liver was a little bit different, not totally unexpected considering the function of the liver. PFOS or PFOS was detected in every single sample of liver. And so as a result, although no PFOS-based consumption advisories were needed for muscle or heart tissue from this area, we did recommend that hunters do not eat liver from deer harvested within a 5-mile radius of the specific location in Northeast Wisconsin. It's called the JCI Tyco facility. This 5-mile radius is estimated to be the maximum travel or dispersal range for deer. That's fairly conservative. Deer probably don't range that far, but we wanted to be conservative on purpose and also to be a little bit compatible with the range that Michigan did, although their advisory is in a different part of Michigan. It is a border location, and so we try to keep things relatively similar. And based on these results, we wanted to determine whether or not the PFOS levels that we observed at that contaminated site were a reflection of local contamination, or if those are simply represent PFAS levels in Wisconsin white-tailed deer. So we recruited 80 volunteer hunters, which were all Wisconsin DNR staff. Samples were collected during our 9-day gun deer hunt, which occurs in late November. We collected a total of 52 samples, and we submitted 32 samples for analysis. We had some duplicates, but at this point, we submitted 1 sample per county. And we expect those results in a few months. And again, the purpose was just to determine what is background and whether or not those results that we observed at that contaminated site were a result of local contamination. So just some acknowledgements with the Bald Eagle project. The project was started by Mike Meyer and our current crew: Dan Goltz, Steve Easterly, Matt Stuber, Brian Dewey, Lindsay Long, and Mandy Camps. The Wisconsin State Laboratory of Hygiene analyzed all the samples. And as I mentioned, USDA Wildlife Services collected the white-tailed deer for us. And with that, I will take any questions, and I will also provide my contact information.
So I was very eloquent on mute. Thank you everybody for speaking, and if I can have all the panelists please turn their video on. I see we have a question in the chat that didn't get answered, and I want to make sure that we touch on questions and open this up to questions that anybody has that hasn't put it into the chat window here. We had a question about, could TEM help determine what is in the hepatic vacuoles? And I think, Mark, that was something that you had been talking about. So we'll direct it to you first, and if others want to chime in, we can do that.
Yeah, I'm not a transition electron microscopist, but I don't think so. I think really what you need are either special stains that can stain the material so you know what it is, or sometimes you can use monoclonal antibodies that have an epitope that's specific to the thing you're looking for, that has a light or target on it that you can see under a microscope. And I just don't know if they have any for PFAS yet for certain molecules. So I think the technology just isn't quite here yet, but it's not that far away.
And then we got a question from Kristen Schuler here. Have other states issued consumption advisories on wildlife because of PFAS?
So far, the only one that I'm aware of is Michigan with their white-tailed deer. I do believe, and I'm not 100% certain, but I think they may have suggested that you don't consume any wildlife from that area. But I believe that is the only other one, although I do know that there are other states that are looking at white-tailed deer and waterfowl.
Yeah, there are fish
Sean,
advisories.
Can we broaden that to fish? What about fish advisories? Because I know Wisconsin has one,
yeah,
as you mentioned.
Yep. There's a lot of states that have issued fish consumption advisories due to PFAS. Absolutely.
Okay, and Diane Kopek notes that in Maine, at least one is warranted next to a military site, but it's superseded by a mercury advisory. So we got another question here. Is the fluoride in these chemicals bioavailable in the body as a toxin?
I guess I'll take
that
at it. This is Mark. I don't think so. Because the problem with halogenated organics is they're persistent, right? And that's because that very, very strong fluorine carbon bond. So yeah, I don't think fluorine's an issue here.
Okay. And then when Sarah Bowman asks, has there been any work on understanding if there may be competing uptake of various PFAS by fish or wildlife that are exposed to PFAS mixtures? For example, at lower PFOS concentrations, could there be more efficient uptake of another PFAS such as PFNA?
Yeah, I don't want to hog the conversation, but man, what an excellent question, because I've seen this with other molecules, right? I've seen it with DEET, which is, insect repellent. It penetrates the skin like a hot knife through butter. But if you mix it with other stuff, we call it compound facilitated transfer. It goes right through skin. It can move other compounds through. Can some PFAS do that? It's plausible, but I don't know if anyone's ever looked at it.
Chris, is there any of your work that can answer that, or is that outside of the realm of what you've been able to get to?
Given that we're a field, it's hard to know what they're being exposed to. We do have some snapshots in time with the diet, but yeah, I don't know that we could tease that out with any of our swallow data.
Okay.
But hopefully Natalie at Patuxent, is thinking about feeding mixtures. So, maybe I think the laboratory would be the place to look at that, just to ask that specific question.
Okay. And going back to the question about wildlife consumption advisories, Brian Hiller added into the conversation here that there is a consumption advisory for ducks in Australia near an Air Force base in Victoria. And he also provided a link there, so to one of the publications. So if you're interested in that, take a look at that comment there. And then we got a question here about Elsie Persico, lives in Virginia Beach, and obviously there's tons of military bases and industrial areas there. Does anybody know about research going on in that area, specifically south of Chesapeake Bay, somewhere around Norfolk and the surrounding areas? Mark, Chris, Amy?
Yeah, I guess this is Mark. I guess what she's asking is, do we know if the drinking water has been characterized or soil has been characterized, sediment with concentrations of PFAS? I don't know.
Yeah, I'm not aware of the work that's specifically going down there. But likely if there is a military base, I'm sure there's at least a site investigation going on. So I'm sure it's in process.
Okay. What about any wildlife research she adds in? She's a veterinarian, so interested from that perspective.
I guess from the Department of Defense, the only wildlife research I'm very familiar with is that from the Strategic Environmental Research Development Program. It's SERDEP is the acronym. And you can Google that and look at the research that's coming out. In fact, some of the work that I showed is from a work unit associated with that program. There's work being done in birds and other species. And I would just advise you to Google SERDEP and look at their website with that and PFAS and see if they can, you can provide more information there on that.
The program is Strategic Environmental Research Defense Program. Is that right, Mark? SERDP.
Almost. Strategic Environmental Research and Development Program.
There we go. Yep. Sorry. All right. So now we get another question. In plasma, PFAS are associated with albumin. What other molecules are they attracted to in storage tissues? Chris, this sounds like this might be one to direct towards you since it's an albumin question.
So, what we're finding is that the highest exposure is in the plasma of the bird. So, that coincides with the albumin. And then, the eggs are another one. Of course, they've got a lot of albumin. So the eggs in the plasma usually have approximately equal concentrations. And then, the concentrations in the whole carcass drop by about 50%. So, but we're finding in our birds that liver tissue, isn't detecting nearly as many or at nearly as high a concentration. So, if I was doing a monitoring sampling, I would stick with either eggs or plasma and stay away from some of these other organs because we're not finding much of anything in the liver tissue.
Okay. Sean, is there anything you want to, from your stuff that might also help answer?
No, other than that, most of our work has also focused primarily on plasma or muscle tissue as it relates to waterfowl or deer. We did do the liver sampling of deer and did find much higher levels in liver versus muscle and heart. But again, that wasn't totally unexpected. We have done some small mammal sampling with both carcass and liver, and we did find, like in mink and muskrat, fairly high levels in the liver. But again, we didn't analyze plasma as a co-sample, so we can't really say how they relate.
All right. And then we have a question here from Ted Robb about what's the nature of the evidence for the goitrogenic or thyroid effects on wildlife? Is it from free T3 levels?
I'm hoping we'll be able to answer that. We've been looking at T3 and T4 in both the thyroid gland itself and in the plasma. The plasma seems to be holding pretty stable, but so we'll hopefully be able to parse that out and have a better answer to that in a few years with more sample size.
Okay. And so then Margaret Martinez has the question about, is there a resource available to find potential sites in our areas regarding potential sources of these contaminants? And she's in particular is in California, but just in general, kind of that question is where, I don't know.
Yeah, I'm not sure, but I would just say, almost look under any rock. I mean, you'll find PFAS in your house. As I mentioned, it's in stained master carpet. It was in some food packaging. It's in Gore-Tex. I mean, this stuff is really ubiquitous. But I guess the question, Margaret, you're really asking is major like industrial sources that could be in the environmental media, such as, again, soil sediment or water. And I don't know. Typically, California is very proactive in things like that and characterizing these sources. So maybe DTSC, Department of Toxic Substances Control, may have some sources of the investigations they're aware of in California. You may want to try Google DTSC and see if NPFAS, and maybe they have a site. But I really don't know.
A lot of states do have, a lot of states do, and I'm just Googling right now, but it looks like the Water Board in California actually has a map of PFAS sites that they're tracking. I'll put the link in the chat, but they're very likely. So I know New Hampshire does. They basically, if there's a drinking water, they basically, if they have PFAS concentrations that they've sampled somewhere, they have that on a publicly available map, and a lot of states are doing that. So yeah, let me just put that in the chat.
So then a question for all of our panelists. What type of advice do you have for wildlife managers that are getting questions about game or fish consumption, or even questions about wildlife effects? Like, where do they start if they're not a toxicologist, but are starting to hear these questions from the general public?
Well, I'll say from a wildlife consumption advisory viewpoint, we're still kind of early in the game. Fish consumption advisories, there's a long history of it. And so in terms of fish consumption, we're a lot farther along. But in terms of wildlife, we've been monitoring contaminants in waterfowl here in Wisconsin for a lot of years. And so we have a little bit of a background to go with. But still, in terms of PFAS, we're still trying to determine, it's like, well, our levels that we use as guidelines for fish consumption, are those still useful for wildlife? And so, we talk a lot with our state Department of Health Services to try to figure out those questions. And then, in terms of wildlife impacts, following the work of Chris, who is really a leader in this area, that's the best advice that I can give, is just to try to keep on top of the subject. And I get asked a lot, are wildlife safe to eat? And obviously, usually the question is yes. But in terms of specific locations, if there is a serious concern, the thing that I always refer to is these are your family choices, and you have to make the best decision with your family. But I will also say that consumption advisories are relatively few, but part of that is because a lot of us are still getting up to speed on PFAS and wildlife as it relates to consumption.
Others want to jump in, answer those questions?
I just want to say I agree with Sean. I think, like I said before, that the cows are out and the barn door's already closed. If we were really going to see some marked effects, I think we probably would have seen them by now if there were obvious ones. There may be a whole lot of subtle effects out there, and certainly immunosuppression is one of them, right? If you were to do some, I don't know, forensic pathology of sick wildlife, you'd probably come to a different conclusion than PFAS exposure unless you specifically measured for it, right? And one other thing I just want to point out quickly is measuring for PFAS is not easy or straightforward. It is difficult. It requires specific equipment, and it's expensive. And they're still trying to work out a lot of methods associated with different matrices. And so we're still learning a lot on how to do the analytical piece, right? And the other thing that I'm sure I have a lot of friends in this category is that getting funding to do this sort of work for wildlife is difficult, right? And so that coupled with the fact that the analytical work's expensive, probably will tell you that we guys still got a long way to go.
Okay. Chris, we got a question here. If you can expand on your comment about T3 and T4 in blood and bird plasma, are there indications it's associated with some PFAS analytes?
So we haven't seen any on a kind of a site basis and an adverse association, but I would say it's still really early and our sample size isn't huge. But I would, put you in contact with Natalie Corona-Rainier because she's the one that does all our T3 and T4. But I'm hoping with, all of these new AFFF sites and our shift to more emphasis on PFAS that we would be able to answer that question, in the coming years.
Okay. And I'm going to throw one more question out at the panelists. What do you think some of the future research or sampling should be in terms of other species or things that we haven't had a chance to look at that we should be looking at? Because even though we say, hey, we don't see a lot of, Mark, you just kind of said, like, we don't necessarily see a lot of major effects. They might be more subtle, but what haven't we looked at? What are our unknowns that we should be thinking about?
One thing that we're looking at is at this boundary between the aquatic and the terrestrial without any terrestrial inputs, not like a crash site or anything. We're trying to see how far away from that contaminated wetland or pond the PFAS exposure might go, looking at, say, house wrens. But the other piece of that would be then to look at the terrestrial ecosystem where there was a spill or an airplane crash, and look at when that the soil is contaminated, what kind of contamination do you have moving up the food web? So I think there's a couple questions there that might be of interest. And I think it's a lot easier for different people, the companies, to clean up a terrestrial site because you can just get in there and dig out all the sediment and replace it with clean sediment. So, if there are sites that are contaminated, they might be able to clean those up. The aquatic often is a little bit more difficult depending on the size of the lake or the stream. And if it's continually fed, like the picture Amy showed with new contaminated groundwater, then, cleaning up that marsh isn't going to do you any good because it's just going to get recontaminated. So anyway, there's just so many questions.
This is Mark. I think if you want to look for a sensitive indicator species of thyroid effects, that would be metamorphosing amphibians. And so I would like to see more work done with frogs and to see, if there's any issues associated with timing to metamorphosis.
Yeah, I would agree. More work on herpetiles is definitely needed. We simply don't know much about that. I am also very interested in the sublethal impacts, and as well as is kind of related to that is trying to figure out which compounds are a concern or maybe not be a concern. Is PFOS a problem? Obviously it's the driver for human health issues, but maybe it's not for wildlife, and we need to start looking at other PFAS compounds. And then from a consumption standpoint, waterfowl certainly has piqued my interest simply because of their interaction with potentially contaminated surface water that I think from a consumption standpoint, I think waterfowl are a group of interest. And I know we're going to start looking at that a little closer here in Wisconsin.
Okay. All right. We got another question here. These are forever chemicals, but can they be thermally or microbiologically destroyed?
Not my area of expertise, but yes, they can definitely be destroyed. I know that when we take soil offsite and dispose of it, it does go to an incinerator. So there must be a level at which it can be destroyed.
All right. Any other questions from folks? All right. Well, I would like to thank our panelists. Thank you very much for the time you've spent preparing and presenting the material. Very informative today. And I also want to thank everybody that that participated. Definitely appreciate the show of interest in this topic, and we're happy as the Wildlife Toxicology Work Group to answer other questions. You can always reach out to me or any of our other board members and seek some guidance or help.